Good afternoon and welcome to CureAccelerator Live for Diversity, Equity, and Inclusion. I'm Barbara Goodman. Thank you so much for joining us today. Cures Within Reach leverages the speed, safety, and cost effectiveness of testing already approved therapies for new indications. We are institution agnostic, geography agnostic, and disease agnostic, and our funding of over $8 million to over 100 different trials and projects has led to over 80 million in follow-on funding for our researchers and trials. Most of the funding we provide are seed stage funds at proof of concept, first in human, or pivotal studies that can provide the catalytic effect for the next larger confirmatory study. We fund trials whether they have philanthropic or commercial uses, and instead we focus on finding viable treatment options for patients with unmet medical needs. Cures Within Reach's current portfolio of funded trials is broad. This includes trials in both adults and pediatrics. It includes a lot of clinical trials as well as some preclinical trials. And it includes mostly drug repurposing trials, but we also capture devices, nutraceuticals, combinations, and others. 75% of our trials in our portfolio take place in the United States, but they're also occurring in 6 other countries around the world. Not surprisingly, half our trials are in the rare disease space, but our disease agnostic approach is seen on the statistics on the left side of your screen. Specifically, in diversity, equity, and inclusion, our clinical trial efforts are in 3 different areas. First, clinical trials to impact health disparities. Second, clinical trials led by US-based underserved racial and ethnic minority researchers. And finally, clinical trials that are led by underserved researchers that are based in low and lower middle income countries. I want to thank our event partner, DIA, and its 2-day Diversity, Equity, and Inclusion event. All of the attendees of this 2-day event are here joining us today, and we welcome you. In addition, Cures Within Reach is very grateful to our many diversity, equity, and inclusion stakeholders who support our efforts in DEI. Our lead partner is Takeda and its Center for Health Equity and Patient Affairs, as well as the Searle Funds at the Chicago Community Trust. Additional thanks, of course, as well to the other philanthropic and industry partners that are shown on this slide. With that, I'm very excited to get started and I'm going to hand it over to Claire Thibodeau.
Thank you, Barbara, and thank you to everybody who's joining us here today at CureAccelerator Live for DEI. I think that we have a great event ahead of you today with some really fascinating and impactful research to share. And I think you as the attendees are going to have a pretty tough decision when all is said and done. So in just a few minutes, we'll get started with our pre-recorded pitches that our finalists prepared in advance. We'll also have live Q&A featuring an expert panel. Once all the pitches and the Q&A is done, that's when voting opens, and that's when you as the attendees really have your moment to shine. We're looking to you to help us select which project will be selected for funding. You'll vote using the engagement panel located on the right-hand side of your screen. Also included in the engagement panel are posters that we've created for each individual finalist. And we encourage you to take a look at those posters, read them over as you are considering your voting options. So 20 minutes after voting opens, voting will close. And at that point, this portion of CureAccelerator Live will be over. However, more to come tomorrow at 9 a.m. We will be announcing the winner of the funding. So please do join us back here using this very same link to find out who was selected. As I mentioned, we're going to have Q&A with expert panelists, and these panelists will help attendees better understand the projects, get a little bit more information, and really gain more insight. These panelists represent research, academia, industry, and philanthropy, and we are very grateful to have them here today. First, we have Stephanie Crawford from the University of Illinois at Chicago. We have Alfred Mays from the Burroughs Wellcome Fund, Jeff Sherman with Horizon Therapeutics, and finally, Kwame Yaboa from Harding University. Thank you all. And just a few things for you, the attendees, to keep in mind as you're listening to these pitches. We do have some criteria that we recommend for voting, and we do ask that you weigh all of these criteria equally. The first is patient impact, the probability of success of the project, the speed to patient, the research team, and the overall presentation. Now, today you'll be hearing some projects that are led by underrepresented minority researchers. You'll also be hearing some projects that impact health disparities, and in some cases, the projects will be doing both. But what's important to keep in mind is that all projects are equally eligible to receive funding from Cures Within Reach. So that's one voting consideration you don't need to keep in mind going forward. And of course, it would not be CureAccelerator Live without our fantastic finalists. And we have finalists from all across the country, literally coast to coast. Not only do we have PIs, researchers from a variety of different institutions, but they're also looking at a variety of different disease areas, including neurology, dermatology, oncology, and rare diseases. So again, we are just thrilled to have them all here joining us today. And I think that instead of me telling you more about them, what would probably be better if we just went ahead and started with our expert or our pre-recorded pitches. The first PI that we will be hearing from today is Dr. Amorim from the University of California at San Francisco, and his project testing the epilepsy drug perampanel to prevent seizures after cardiac arrest. Take it away, Dr. Amorim.
Hi, I'm Eddie Amorim. I'm a neurologist at University of California, San Francisco, and director of the epilepsy program at the San Francisco General Hospital. My clinical and research career is focused on treating seizures after cardiac arrest, and I'm confident that by the end of this presentation, you'll see the potential of perampanel to improve patients' lives. This is a patient story that had a transformative effect in my career. A 43-year-old man was playing racquetball when he collapsed. His heart stopped for 30 minutes, and after CPR, he was in a coma. Soon after his arrival to the ICU, he started to have very severe seizures. Most doctors thought he had irreversible brain damage and that further interventions were not likely to help him. Fast forward 9 days, and he wakes up and holds his daughter's hand. This man who was not supposed to survive went to have a full recovery and go back to work as an engineer. This only happened because his doctors treated his seizures aggressively. This story, this patient, taught me not to lose hope. But why this matters? More than 500,000 of Americans have a cardiac arrest every year, and 100,000 survive to the ICU. 2/3 of patients die in the hospital, most of the time from brain injury and not from heart damage. And up to 1/3 of patients have seizures, and near all patients who have seizures die. Black and Hispanic patients are hit particularly hard, and mortality is twice as high. In the San Francisco County Hospital where I work, almost half of the arrests are related to substance use. We see more fentanyl overdoses than heart attacks. But let me tell you what the patient journey is. People who survive CPR and get to the ICU have EEG monitoring started as soon as possible to monitor for seizures. Most seizures start within the first or second day after the cardiac arrest. But the current practice is reactive, and doctors wait for seizures to start to give medicines that can stop seizures. If I tell you that you have a 33% chance of being hit by a car when you're crossing the street, I'm sure you would find ways to prevent that from happening. And that's what we're trying to do, prevent seizures by starting treatment early. We want to use perampanel, Perampanel is the only anti-seizure drug that is predominantly working in AMPA receptors. These receptors can prevent seizures, but also prevent depolarizations. The slow brain waves decrease blood flow in the brain and can trigger more seizures. In this video, we see a mouse's brain where the light triggers the depolarization. We can observe how there is loss of blood flow in the brain surface. We want to stop this mechanism with the use of perampanel. We have already recruited an experienced research coordinator to help with this study. With funding from and support from Cures Within Reach, we'll be able to start enrolling patients as soon as January in 2023. We are the busiest hospital in the city of San Francisco with near 250 arrests a year. We plan to enroll 3 patients per month and reach our goal of 30 patients enrolled by October. With this pilot completed, we'll be in a good position to get a larger clinical trial funded by the NIH in 2024. But what's the study going to look like? As soon as patients get to the ICU, they will be randomized to perampanel, 1 pill a day, or placebo. We'll do that for 7 days. We'll enroll 15 patients per study arm. The primary endpoint will be development of seizures, but we'll also look at brain injury on MRI and functional recovery. And I'm not going to do this study alone. We have a very experienced team of investigators. I have participated in more than 20 clinical trials. Dr. Ferrero is a leader in the field and has done clinical trials specifically in hypoxic ischemic encephalopathy. Dr. McCulloch is an experienced statistician who will help us make the most of this pilot study. Importantly, we already hired an experienced clinical trials coordinator. But to keep her in our team, we need your help. As I said, most of the funding will cover our research coordinator. A small portion will be towards medication, purchase, and activities with our community. I have already secured funding for a clinical trial in cardiac arrest and seizure treatment with a foundation, and my salary is already supported by the NIH to do research in this area of cardiac arrest and seizure. So why this project will have an impact? This study only involves taking 1 pill a day for 7 days, something we already do in the ICU for all patients with traumatic brain injury to prevent seizures. We can have this study completed in a year by the end of 2023, and we want to start a more definitive multi-center study in 2024. Our team is very experienced in clinical trials, and we have the expertise in cardiac arrest and the statistics to have the right study design to make the most of this relatively small pilot study. Our hospital serves an underserved population who is disproportionately affected by this disease, and we have more than 250 patients a year, so we're in a good position to enroll sufficient patients in just a year. And remember, cardiac arrest is the 3rd cause of death in the United States, and we could advance the care of more than 100,000 Americans every year. Through this smaller pilot study towards larger clinical trials, we're anticipating a 30% treatment response with seizure treatment and prevention with perampanel. This could lead to a return of investment of $100 million per year in the United States alone. And remember, 90% of patients who survive a cardiac arrest can have a good outcome, so I know we can have a big impact. It is my life commitment to help this 100,000 Americans who suffer cardiac arrest every year. I want them to have the same happy ending that patient I talked to you about early in this presentation. Today, I count with your support to make this happen.
Thank you, Dr. Omorim, for that presentation. That's an issue that I think many people may not think about until they really have to. So now I'd like to bring in our expert panel to begin our Q&A, our first Q&A session. Let's see, Kwame, do you want to ask the first question of Dr. Omorim?
Yeah, Dr. Morin and your group may congratulate you on a wonderful project that you come out with. My question is that the seizures cause a lot of death, and the high prevalence of mortality, high the moment they get a heart attack and they get seizures, the chances of death is high. That's why you are coming out with this wonderful thing. You also know that the drug lasts in the body. Half-life is over 100 hours. So by the time, if the seizures start in 3 days, 3 days, over about 72 hours or so. So by the 5th day, we would have known who is benefiting and who is not benefiting. So the blinding may not be necessary after 3 or 4 days. Is it possible for you to break the blinding and then allow those who are in the control group to also receive the drug, or maybe use retrospective historical controls?
Thank you, Dr. Boa. If I understood the question correctly, your question had to do with patients who develop seizures and using the drug in this patient group. Is that correct?
Yeah, we know that you are going to use it by 72 hours. Seizures start, and seizures can lead to death. If the drug works well, then it means that by the 5th day, that we will know that these people are benefiting, these people are not benefiting. When that happens, can you break the blinding and allow the controls to benefit so that you can use retrospective controls?
That's a very important question and very important point of clarification. So the design of the study is to prevent seizures from happening. As soon as someone has seizures, they're going to go into the standard pathway of treatment, which include using anti-seizure drugs. So this will not affect the standard of care. So this is very similar to traumatic brain injury. That's what I take care of those patients in the ICU. Everyone gets seizure medications as soon as they get to the hospital, and we keep that for 7 days. But if someone develops seizures, they're going to have the treatment. The same thing would happen here in cardiac arrest. If you have seizures, you will have treatment. So that part of the care is not changed. We are trying to prevent seizures from happening, and perampanel can not just prevent seizures, but there are different mechanisms like the spreading depolarization potential for prevention that might help recovery of patients after brain injury.
Great, thank you. Stephanie, do you have a question for Dr. Amorim?
I do. Thank you so much. First, I appreciate that clarification because that was my first question too, but no fear, I have another one right ready. I appreciated when in the slides you were talking about community engagement. Dr. Morum, I asked that you give us an example of what will be involved in the community engagement and tell us why is that important in your study.
Absolutely. So I work very closely with the fire department here in San Francisco, and we have specific neighborhoods where we have a much higher chance of patients at risk for overdose. So I think it's very important for us to engage this community to prevent overdose from happening. That's something that already happens with naloxone distribution. But one thing that might not be top of mind for those patients in that community is that what happens other than giving naloxone if their pulse stop in terms of life support and basic life support. So we're engaging that type of education already with the fire department. So I have regular meetings with them with this goal, because unfortunately, that's a really major problem here in San Francisco, downtown San Francisco particularly.
Thank you.
Great. Thank you. And Dr. Amorim, thank you very much for your pitch. We are out of time for the Q&A, but we will be moving on to our next finalist at this point, Dr. Gebra and his project, a new use of the antacid drug esomeprazole to treat morphea, a chronic connective tissue disease.
Hello, and good afternoon. My name is Johannes Gebre, and I'm an associate professor at Baylor College of Medicine. Today, I would like to talk to you about the potential of repurposing the FDA-approved antacid drug esomeprazole for the treatment of morphea. First thing first, what is morphea? Morphea, also known as localized scleroderma, is a rare autoimmune disease characterized by excessive inflammation and abnormal deposition of collagen in the skin and underlying soft tissue. As a result, it causes pigment change, as you can appreciate in the image. In its severe form, morphea can also cause impaired mobility, including joint contractures, as shown in the lower panel. How common is morphea? Currently, the disease has an incidence of up to 3 cases per 100,000, and it is estimated that there are about 30,000 patients in the US alone. It is important to note that women are disproportionately affected by morphea at 2.6 to 1 ratio compared to men. Although the cause of morphea is largely unknown, new onset of the disease in the settings of radiation therapy has notably increased over the years. The image shown provides an example of radiation morphea. So, what is the current treatment for morphea? It is unfortunate that there is no curative pharmacotherapy for morphea. However, immunosuppressive drugs are used to manage the disease and alleviate symptoms. Unfortunately, the use of those agents is associated with significant side effects, including toxicity and risk of infection. It is intriguing to note that none of the current treatments for morphea target fibrosis. This is despite the fact that pro-fibrotic pathways are upregulated in morphea at preclinical and clinical settings. Sadly, there are not very many candidate drugs on the horizon. Of the very few that are in clinical trials, most of them are immunosuppressive drugs targeting inflammation, and none of them targets fibrosis. Well, what if I tell you that there is a generic drug widely available in the market that can simultaneously target both inflammation and fibrosis? Please meet the FDA-approved anti-acid drug, esomeprazole, also known as Nexium. Esomeprazole was identified to have anti-inflammatory and anti-fibrotic activities upon high-throughput screening of 130,000 small molecules against an enzyme known to drive inflammation and tissue fibrosis. We then formulated esomeprazole into a topical product, which we named dermaprazole, and assessed its anti-inflammatory and anti-fibrotic potential in a mouse model of radiation-induced dermal inflammation and fibrosis. As the data shows, dermaprazole is a very effective anti-inflammatory and anti-fibrotic product. This is shown by the accelerated healing of the irradiated area in the animals that receive dermaprazole in a prophylactic or therapeutic manner, as shown in the last 3 data sets. From the data, you can also appreciate that the corticosteroid-treated animals still show necrotic skin. I don't have the time to show, but we also have positive data in an animal model of localized scleroderma, where the dermal inflammation is chemically induced by bleomycin. So, what are we proposing to do? We're proposing to conduct a phase 1/2A clinical study that is primarily focused on safety and tolerability of dermaprazole in patients with morphea. We are using the standard 3+3 study design to first conduct patch testing in healthy volunteers to assess the safety of 1% and 2% dermaprazole over the course of 6-week treatment, followed by 4 weeks of follow-up. If found to be safe, we will then proceed to patch testing in an unaffected area in morphea patients using the maximum tolerated dose identified in the above studies. If found to be safe and well tolerated, we will then enroll 12 patients with morphea to conduct a 3-month safety and preliminary efficacy study. Here are our endpoints. For safety, we are looking at burning, itching, tenderness, rash, and/or allergic reactions to dermaprazole. For the efficacy study, we will use 2 well-validated tools to assess the disease activity and quality of life. One may ask, what would the proposed study cost? We are asking for up to $70,000 in funding. Most of this funding will go towards study personnel, drug purchase, and formulation of the product for 30 or more study subjects, including the patch testing. Remember, the drug is only available in oral and IV forms in the market, and it needs to be formulated for this study. Who is involved in the study? I am translational researcher and study PI. My lab has pioneered the dermaprazole technology. Dr. Jacobi is a co-investigator She's professor of dermatology and exclusively sees morphea patients. Dr. Myers is a project advisor. She's internationally recognized expert in scleroderma research and clinical care. Dr. Ramamurthy is a co-investigator. She's an expert bioinformatician and biostatistician. She has several years of experience in data coordination for clinical trials. To summarize, morphea is a rare and understudied disease that affects humans across lifespan. It is lamentable to know that there is no single pharmacotherapy that can treat morphea. That's why you have the opportunity to support the first in-human clinical trial to repurpose a widely available generic drug for the treatment of morphea. Supporting this study presents an unparalleled opportunity to leave a lasting legacy in the lives of morphea patients, their families, and the community at large. With that, I would like to thank you and happy to take questions.
Thank you, Dr. Gebra. One of the things I find interesting about morphea is that it really does cross a number of different disease categories: rare, dermatology, and inflammation. Turning back to our expert panel now, Alfred, would you like to ask the first question for Dr. Gebra?
Sure, thank you. Dr. Gebra, with a ratio of 2.6 to 1 between women and men, is there any insight into why women are affected so disproportionately? And will the trial have any element of examining this based on subject selections or reactions to treatment based on gender?
Hey, thank you, Alfred. That's very good set of questions. So unfortunately, it is not clear in the literature why women are more susceptible to getting morphea compared to men. So that is not known, and I don't want to speculate to something that has not been figured out. But that's an important question that the basic science research is still investigating to find out. But we will certainly include women in our study, in our clinical trials, and see how they respond compared to men, because they may be more susceptible to the developing morphea, but would they be less responsive or more responsive to treatment? That is also an important thing that we can control in our study. We will be able to address that. I hope that answers.
Thank
it does.
Kwame, do you have a question for Dr. Gebra?
Yeah, Dr. Gebra, this is a nice project. Now, why did you choose patch? Because patch has a different dimension. You don't use big patches. This thing affects women most of the time. Why don't you use ointment-based or cosmetic-based delivery system? We added up, and the disease is spread over the skin. They can use cosmetic-based delivery system to deliver the drug. For the safety, I understand, but the efficacy, why do you choose patch?
Yeah, thank you, Dr. Eba. A very good question again. So the patch testing is only for the allergy test or for the initial testing, but for the actual testing, we will not be using patch. You're right that the disease could be spread all over the body, so they will be freely, able to topically apply this product. So the patch testing is just basically out of caution to restrict the area of application, so that in case there is a reaction to this topical, we'll be able to monitor that.
Okay. Yeah.
Thank you. Jeff, we haven't heard from you yet. Would you like to ask a question of Dr. Gebra?
Yeah. Can you really tell us about, this is a unique formulation of esomeprazole, which is usually given orally. What's been done to really test this? As I see, it's a topical formulation, unless I misread something.
Yeah, that's correct, Dr. Sherman. So this is a topical formulation that we formulated in collaboration with a compounding pharmacy based in here in Houston. So it is a lipoderm-based topical product that is able to penetrate the skin because lipoderm, as you know, has been used cosmetically. But also there's a different product called vanishing cream to restrict it from being released to the bloodstream, so that the drug is mostly going to stay in the dermis, which is where it's needed. But also this gives an additional layer of protection in terms of intellectual property, because cannot be used off level because it would not be available, because the topical application is to be available by prescription only, not, as you said, the commercial products are oral formulation only. So I think it provides additional layer of protection for the IP, as well as to minimize systemic exposure for the patients, because you could directly apply it topically to the area that's affected.
Thank you.
Thank you.
Great. Thank you again, Dr. Gebra. We really appreciate your presentation and your efforts to help patients.
Thank you.
Next up, we have Dr. Sheldon Holder and his presentation on repurposing the prostate cancer drug Degarelix to treat bladder cancer. Dr. Holder?
Good afternoon. Thank you for the opportunity to present our clinical trial, Repurposing Degarelix to treat bladder cancer. Bladder cancer is a common cancer in the United States with over 81,000 new cases annually. Approximately 50% of those cases will be muscle-invasive bladder cancer. We treat muscle-invasive bladder cancer aggressively with chemotherapy, followed by surgery to remove the entire bladder. Unfortunately, about 30% of patients, even after this aggressive therapy, will develop distant recurrence. That is, they will develop bladder cancer outside the bladder. And you may ask, how is this possible if patients have had chemotherapy and then have their bladder surgically removed? In these patients, they actually initially present with what we call micrometastatic disease. That is, disease outside the bladder that is too small to be seen, even with modern imaging like PET scans and MRI scans. And in fact, the chemotherapy that we give is designed to kill the micrometastatic disease. So you may ask, how do we know how well the chemotherapy works if by definition you can't see the micrometastatic disease? We can determine how well the chemotherapy works to kill the micrometastatic disease by looking at how well it works to kill the disease we can see, that is the tumor that is in the bladder. So the tumor in the bladder acts as a surrogate of what's happening to the micrometastatic disease. In patients who have chemotherapy followed by surgery, and at the time of surgery still have muscle-invasive bladder cancer found in the bladder, they have an overall survival of approximately 3.7 years. In contrast, patients who have chemotherapy followed by surgery, and at the time of surgery, no tumor is found in the bladder, those patients have what we call a pathologic complete response and have an overall survival of approximately 13.6 years. So here we identify the problem that we seek to solve with our clinical trial. We need to improve the efficacy of chemotherapy such that patients will have longer survival. Bladder cancer is 3 times more common in men than women. We and many others believe this is contributed to by testosterone. So it is important to remember that testosterone binds to the androgen receptor, and then the androgen receptor moves to the nucleus and causes new proteins to be made. We know that bladder cancer has androgen receptors. We know that androgen receptor signaling promotes chemotherapy resistance. We also know that men who are being treated with testosterone-lowering agents for their prostate cancer have a lower incidence of bladder cancer. And we know that men who have both bladder cancer and prostate cancer and are on testosterone-lowering agents for their prostate cancer have a lower recurrence rate of bladder cancer. In the Holder lab, we have also shown that if you treat bladder cancer cells with chemotherapy in the presence of testosterone, in the bottom left circle, you can still see that those bladder cancer cells are alive. But in the bottom right circle, circled with the green circle, you can see that if we treat patients with chemotherapy in the absence of testosterone, those cells are dead at 3 days. Again, showing that by removing testosterone, you improve the effect of chemotherapy. This is exactly what we propose to do in our clinical trial. Patients who are receiving chemotherapy for their muscle-invasive bladder cancer will also receive a drug called Degarelix. Degarelix dramatically lowers testosterone levels. It is FDA approved and standard of care therapy for prostate cancer. We believe that by adding Degarelix to chemotherapy, we will improve pathologic complete responses. Patients in our clinical trial, we will enroll patients with androgen receptor positive bladder cancer. The primary outcome will be the pathologic complete response rate. Historically, this is only 35%. We believe we can improve upon that. An important feature to remember is that treatment is only 3 months for the chemotherapy and the diuretics. So for each individual patient, we will arrive at the primary outcome at only 3 months. Additionally, this is a pilot study. We only require 32 patients. And so we believe that with a short time to the primary outcome and only requiring 32 patients, this is a clinical trial that can be completed in a relatively short period of time. I'm showing you here our budget. You can see most of the budget is budgeted for purchasing the diuretics. I do want to note, second from the bottom, that we have also budgeted for patient assistance. We know from prior history of clinical trials that non-medical issues, social issues such as unable to obtain transportation can be a barrier to being able to enroll patients into clinical trials. And so we have budgeted to be able to assist patients with these social barriers to enrolling in clinical trials so that we can assure that we are able to enroll 32 patients required. Additionally, our department at Brown, the oncology department, is also excited about this clinical trial, and they have done a rare thing in committing $40,000 to this study so that we can have the total amount we need when adding to the $70,000 from Cures Within Reach so that we can conduct this study. I'm showing you here the clinical team. I am a physician scientist. I run a translational cancer research lab, and I'm showing you the lab members on the left. And I also treat patients in the clinic who have bladder cancer in a multidisciplinary clinic, and I'm showing you the other physicians on the right. Additionally, we will conduct the study with the Brown University Oncology Research Group, which has conducted clinical trials in over 80 types of cancers. And some of those clinical trials have led to practice-changing national studies. For our proposed study, if we see efficacy, and we believe we will then plan a phase 2 trial, which will be randomized in patients with muscle-invasive bladder cancer. This is the industry standard for showing that a new therapy is improved over an old therapy. But not only that, we will also plan a phase 2 trial for patients with non-muscle invasive bladder cancer and patients with metastatic bladder cancer. Thus, this proposed clinical trial will have depth and breadth of impact with over 700,000 people living in the United States with bladder cancer. Thank you for taking the time to listen to our pitch. We hope we can garner your vote to obtain funds to perform this very important clinical trial.
Thank you, Dr. Holder. Your project is a really great example of how one cancer drug can be useful in treating one or multiple other types of cancers. So Jeff, would you like to start the questions from our expert panelists?
Sure, thanks. And thanks again, Sheldon, for a great presentation. My question is really about, dosing. You're anticipating the same dose that's used in prostate cancer or not? Any questions or concerns since this will be added on to standard chemotherapy? If you can describe what that standard chemotherapy is that you use at Brown, any questions or other, concerns about, toxicity, because it's always about benefit risk for patients.
Sure. So we do plan to use the standard dose for our clinical trial that we use for prostate cancer. And we don't anticipate increased toxicity, although we'll be looking for it. But the hormone suppression from the diuretics can cause usually hot flashes, maybe some fatigue, but we don't really expect overlap with the standard side effects from chemotherapy that we use. So we typically use a drug called gemcitabine and a drug called cisplatin for chemotherapy. They don't really have overlapping side effects or toxicities, except for maybe the fatigue. And so we will be looking for overlapping toxicity, but we really think it'll be a very tolerable regimen by putting the two together.
Thanks.
Thank you for the question.
Alfred, what question do you have for Dr. Holder?
Thank you for a great presentation, Dr. Holder. Your presentation notes the reduction in testosterone, and I'm curious about the reactions by primarily the male patients. And what do you foresee as the communication around a patient's understanding of the process and the side effects?
Well, fortunately, we use digorelic's or drugs similar to digorelic's very frequently to treat men for prostate cancer. And so I'm very familiar with having that conversation with them. And we usually give them a good conversation about what side effects to expect. Again, hot flashes is very common. And although men will have side effects, it's very uncommon for the side effects to be so great that men would discontinue the therapy. And so we would do the same thing here. We would give them a very good discussion about what side effects to expect, but we don't really think that there will be dose-limiting side effects.
Great, thank you.
Thank you.
Stephanie, let's hear from you.
Thank you, Dr. Holder. Just a quickie clarifying question. The inclusion criteria includes patients with androgen receptor positive bladder cancer. Please clarify, will only men be enrolled, and why or why not?
Oh, great question. Thank you. So yes, we will only include patients who have androgen receptor positive bladder cancer. However, this will be men and women. So it's been well documented that women who have bladder cancer are just as likely to have androgen receptor positive disease as men are. And not only are they just as likely to have androgen receptor positivity, they're just as likely to have high androgen receptor as opposed to low androgen receptor as men. So
Thank you.
I think the only difference will be just the difference in gender that we see up front with women having less bladder cancer than men do. But the percentages being androgen receptor positive should be the same.
And Kwame, we do have time for one quick question, if you have one for Dr. Holder.
Yeah, I was thinking about the cost of this combination thing. This happens in the institutions. The cost to third world countries, if underserved communities that need this treatment to survive. What do you think would be the cost? How are we going to be able to send this around the world for their benefit?
That is a good question. So we're using digorelic's because it's available as an injection that lasts for approximately a month. There are other drugs that may be less expensive that can be used to lower testosterone levels, but they don't lower the level as fast. So they would tend to lower the level in weeks, as opposed to the injection, the digorelic's that we're using, which will lower the testosterone level in days. Up to 3 days is usually the time to greatest to lowering the level. And so I think that one thing that will actually hopefully decrease the cost of the drug is when you're able to use it for more indications. So right now, digorelic's is only FDA approved for prostate cancer. So potentially there would be a decrease in the cost of the drug when it's being used for more indications.
Great. Well, thank you, Dr. Holder. We really appreciate your time today. We will now be moving on to our final presentation of the event, Dr. Schur and her project, using steroids to improve outcomes in rhabdomyolysis in pediatric patients.
Thank you so much for the opportunity to present our project, Rhabdomyolysis, a need for evidence-based interventions to decrease health disparity and improve standards of care. I'm Dr. Natasha Schur, a medical geneticist at Children's National in the DC area. Some of you may have heard of rhabdomyolysis, a disease of muscle breakdown associated with spin cycle classes or marathon runners, but in fact, many children, adolescents, and adults come into the hospital after a virus. Some have genetic disorders, others it's out of the blue, and these kids are in severe pain. pain in their legs, chest, abdomen. They experience muscle cramping due to muscle injury. And this disease can cause severe kidney damage and even be life-threatening. In recent years, we noticed something very unusual about our rhabdomyolysis cases. This disease has been disproportionately affecting communities of color. For example, at Children's National, over 50% of patients who come in with rhabdomyolysis are reported to have Black race. Only 17% are Caucasian. And nationally and internationally, there is a twice as much increase as expected of Black race and other patients from communities of color. There are over 26,000 hospitalizations with rhabdomyolysis per year, and this disease is increased with sickle cell trait and escalating with the COVID-19 pandemic. Unfortunately, treatments are very limited, just IV fluids and supportive care. More has been done in racehorses, where they try a medication called dantrolene before the race to decrease the chance of rhabdomyolysis. And the military is studying agents that decrease the risk of kidney function. But meanwhile, patients who are diagnosed, and the 1 in 10 with rhabdomyolysis that go on to have many recurrent admissions, face severe exercise restrictions. I've seen kids who've had to quit football or crew because of this, whose lifestyle, whose future is changed. I think that we can do better. And many years ago, I had a very sick 4-year-old with a rare metabolic condition that caused rhabdomyolysis, and his CK, a measure of muscle breakdown, was rising. He was in pain. I was extremely worried, and I called people around the country until one expert said, try some steroids. And when I did, and the steroids worked remarkably well, this inspired me to read more about the pathophysiology and try this approach again. And over time, we published a protocol and our experience with a few patients where steroids have been successful. And I want to share one patient's mother describing her particular case.
Dr. Shore and the team kind of mentioned starting some steroids to see if we can get the numbers down, because his numbers were like triple the number in his CK level that it was supposed to be. So it was a really scary situation, and my mom's in a lot of pain. And so Dr. Shore and the genetics team came in and started the steroids. We noticed once the steroids started and got really good into this system that pain went away, the numbers went down. It was just like, oh my goodness moment for us.
And in thinking about her case and how not every child receives this treatment when coming in, we decided it was time to do something different and perhaps try a trial where we chose steroids. In this particular case, we chose a steroid called dexamethasone because it's familiar, it's been used in pediatric asthma protocols, it's cheap and accessible. However, uptake of trying this novel approach has been limited. And in thinking about why, I wanted to describe the experience of a team member on a recent case. Why are physicians hesitant to try this?
I remember walking into pre-round the following morning after seeing my patient, and I heard the attending sharing with the senior resident, well, genetics team actually suggested dexamethasone. I heard dexamethasone, what? And the attending just goes, yeah, I don't know, but we consulted them. So we shrugged our shoulders and went ahead and did that for that day's plan. And the next day was the first day this patient's CK went down after continuously rising over 100,000 over a week, even with adequate IV fluids. And it was the talk of the next morning's rounds with relief and excitement and curiosity. So it's my impression that rhabdo has not been well talked about, especially based on the discussions my team had about the disease, also attributed by the limited exposure these physicians have had to it, let alone during my preclinical years of medical school. So the success in symptom relief was really incredible from dexamethasone.
In order to overcome that natural hesitancy to change decade-old protocols, we really need more data. And because of this recent case, so many physicians were garnered to help us to say we want to try something new at Children's National, one of the first pediatric trials for rhabdomyolysis, this underserved condition. So we have my colleague, Dr. Berger, who's a great researcher and statistician. We have emergency room physicians, we have hospitals, and a research team on board. We've already prepared a protocol. It just needs to be tweaked. We're looking at 10 years of natural history. We would begin active recruitment within the first 6 months. Community partnerships will be key, and we plan to analyze the data of dexamethasone along with standard care versus standard care alone within 3 years and publish our findings. Most of our budget would go to a research assistant who was on site and available to help both the hospitalist and the emergency room setting. And of course, there are other costs with any clinical trial that are fairly standard. But in thinking about our opportunity, we feel that this cheap, accessible, new repurposing trial could help us overcome the hesitancy of trying something new.
Thank you, Dr. Schur. I especially appreciate you mentioning the importance of clinical trials, such as the ones that are being presented today, in changing clinical practice for unsolved diseases. So for our final round with the expert panelists, I'm going to ask Stephanie to start with the first question.
Thank you. Dr. Schur, I appreciated how you used all of the time for the video. I'm going to ask and give you the opportunity to give us just a little bit more detail about your proposed methods, such as the number of study participants you plan to enroll, and how will you determine the dosage of DEX, or any other way you want to take that?
Thank you so much for those questions. So we used Trinetics, and we also looked at natural history data over the last 10 years, and we anticipate about 5 patients per month coming in who have rhabdomyolysis. But of course, we need community engagement. We need to get the word out there, because I'm worried that not all 5 will want to participate. So we've already started going to the ER, the hospitalists. We want to use this video, get more videos, so we can get as many of those 5 per month signed on.
Thank you. And to determine the dosage of DEX?
Oh, yes. And to determine, and to determine the dosage of dexamethasone, we used the standard asthma protocols, and we just wanted somewhere to start. And then we've done that on so many patients since this time that we're not officially part of a study. We couldn't collect the data, and it has been fine. So that's the way we did it.
Thank you.
Great. Perfect. Jeff, do you have a question for Dr. Schur?
Sure. You know, there are a number of different steroids you could have chosen. So I have a couple of questions for you. Why Dex versus something else? And then, you know, in going over the proposal, and thank you again for the great video, as well as the poster. You talk about, in addition to standard of care, if you can describe that and how you're going to follow these patients, because we know one of the potential complications from muscle breakdown is kidney damage.
Yes, absolutely. So in terms of question 1, when I first tried this, somebody had given advice. I used prednisone, but then when I went to our hospital ER people at Children's, our hospitalists, and begged them to try it again, my team did, the genetics team with our protocol, we asked them what they would be most comfortable using. And so our ER physicians and hospitalists said, I guess dexamethasone, since we use it all the time. So it was kind of more just from feedback. And then, but there was no study we could use to determine which drug is best. That could still be studied. And then, I'm so sorry, could you repeat your second question?
Yeah, the second one had to do with, you know, standard of care, monitoring these patients,
Yes.
given, you know, one of the complications of the muscle breakdown is possible.
Exactly. As kidney failure. So right away, when they do have kidney failure, it's usually early in the disease process when they're acutely ill. So if they're hospitalized, they are not discharged until there's some sort of plan with the kidney failure. The hope was that the dexamethasone would decrease the 40% risk of the renal failure. However, in our study protocol, we have something where we had hoped to do a telemedicine visit 14 days out, check in on people, and they'll also check in on their comfort with the plan, both as part of the study, but we are also encouraging that on a clinical basis for every patient with rhabdomyolysis as well.
And then I noted the telemedicine, but then how often are you going to be checking their labs for renal function?
So most people, if they do well with the acute episode, they need to know that it can recur. It's a lot like asthma. If you have an exacerbation and it recurs, that's the time to really check things again. But I would not anticipate, unless they had initial kidney damage in the hospitalization, that they would suddenly have it without an episode again. For those that had kidney damage, they have to be followed by the kidney specialist. And I would assume, depending on the extent, whether it's every 3 months or 6 months or a year, depending on the extent of their kidney damage, is my understanding.
And then the age ranges, you said 0 to 21. That's a pretty wide age range.
Mm-hmm.
And then are you looking for the dex to be administered orally?
Exactly.
And then what about for young kids who may not be able to take it orally?
Yeah, so we found something so interesting. There's a bimodal distribution published in the Journal of Pediatrics in Korea before, but we want to publish it and are in process of writing the manuscript. So we see a peak early on at around 2, those people are more likely to have maybe something genetic or wrong. And then we see another peak between 10 to 12, and this has been again published before. So we wanted to make it broad in terms of the ages, so we're not excluding groups. And then in terms of the dexamethasone, the PO versus the IV, that's another reason I should have mentioned that we chose dexamethasone. I wanted something that we could use very easily and interchangeably. And that way, if somebody is too sick to tolerate a medication, we can use it IV. If they are well enough, they can get it by mouth. And my hope is that eventually, like I've been doing on the patient whose mother had the video, I now treat him outpatient with the dexamethasone, and he doesn't come in anymore. He's only come in once.
Great. Well, thank you, Dr. Schur. I'm afraid that we are out of time, but I appreciate your video and the video of all the other finalists this afternoon. And also thank you very much to our expert panelists for all of your insight. Now I'd like to turn it back over to my colleague, Barbara Goodman.
Wonderful. Thank you so much, Claire. And again, amazing job by all of our finalists. And thank you again for our expert panelists. We're very excited to reach this stage of our event tonight. And that's where we turn it over to all of you, the attendees, who have an opportunity to vote. So voting is now going to open. If it's not open at this split second, give our platform just a second to turn it on, but it'll be opening any minute. As Claire said in the beginning of this session, please do keep the following things in mind when you're considering which of the finalists project to vote for: patient impact, probability of success, speed to patient, assessment of the research team, as well as overall presentation. As we said, voting is going to open it's open now, and it's going to be open for 20 minutes. In 20 minutes, so at approximately, let's see, it's 4:20 Central, 5:20 Eastern, 2:20 on the Pacific West Coast. It'll be open for 20 minutes. And please do turn in tomorrow morning when we will announce the winner. Same link that you went to today, and we'll announce the winner tomorrow morning. With that, I want to again thank our DIA partner and the 2-day DEI event, as well as thanking our diversity, equity, and inclusion partners that are listed on the slides you'll see in a second. Without the support of our donors and our philanthropic partners and industry partners, we wouldn't be able to have a winner, and we wouldn't be able to do what we do, which is all about patients with unmet medical needs. So thank you all for tuning in. Please do get your votes in. You have 20 minutes to consider who to vote for. Go back to the event menu in the engagement panel of the platform, and you'll see how to vote. So thank you so much. And in 20 minutes, we'll close voting.
Thank you.
We're live for diversity, equity, and inclusion. We are really excited to be announcing the winner from last night's competition. As a reminder, if you weren't able to join us last night, Cures Within Reach leverages the speed, safety, and cost effectiveness of testing already approved therapies for new indications. We're a disease, geography, and institution agnostic, and we're really excited to have 4 finalists presenting last night and the results today. Our diversity, equity, and inclusion efforts include clinical trials in 3 areas, impacting health disparities, as well as trials led by US-based racial and ethnic minority researchers, and finally, clinical trials led by underserved researchers that are based in low and lower middle income countries. I want to thank our event partner, DIA, and the 2-day DEI event going on right now, and yesterday as well as our other many stakeholder partners for our diversity, equity, and inclusion community, both Takeda and its Center for Health Equity and Patient Affairs, the Searle Funds at the Chicago Community Trust, as well as the other philanthropic organizations, industry partners, and individual donors that support our diversity, equity, and inclusion efforts overall. With that, let's get to the fun part, why we're all here, and I'm going to turn it over to Claire.
Thank you, Barbara. And again, big thank you to all of our finalists who did such a great job yesterday, Drs. Amorim, Gebra, Holder, and Shore. Again, all of these clinical trials are really important for patients with unmet medical needs and all deserve to be funded. However, as we all know, there can only be one CureAccelerator Live for DEI winner. Or maybe there can be 2 winners. So let's get right to it and find out who they are. The first winner is Dr. Amorim from UCSF for his project looking at seizures in patients with who have experienced heart attacks. And our second winner is Dr. Holder from Brown University and his project looking to repurpose a prostate cancer drug in bladder cancer. So congratulations to both of you. I'd first like to bring Dr. Amorim onto the screen and say congratulations to you. And how does it feel to be the winner or a winner?
Thank you so much for the opportunity. And thank you, Cures Within Reach Foundation, for supporting this work. You know, with the epidemic of cardiac arrest and opioid crisis, there are so many patients in need of help, and I'm really committed to that mission. So thank you again for supporting this work, and thank you to my partners at UCSF and all the other participants.
Congratulations.
Thank you. Thank you. We look forward to working with you. And now let's bring Dr. Holder. Dr. Holder, it's so fun to be able to announce a bonus winner. Would you like to share a few words?
I am very excited and humbled and thankful for this opportunity. And thank you to CureAccelerator, to Cures Within Reach, to the philanthropists and the philanthropic organizations, the expert panel, and the attendees for making this possible. We really hope that we are able to change bladder cancer therapy for the better for an innumerable number of patients. So thank you very much.
Great. Thank you. And thanks again to all of our finalists who, again, did such an amazing job.
We will continue to fundraise and hopefully we'll be able to fund additional finalists within the next few months. If anyone listening is interested in helping us, achieve more finalists to getting grants and getting these clinical trials underway, please do reach out to Cures Within Reach. We look forward to talking with you. With that, we want to thank all of our attendees for joining us last night as well as today. We want to thank our expert panelists, and first and foremost, and most importantly, I should say, we want to thank all of our finalists. You all live with patients with unmet medical diseases and unmet medical needs every day. We wouldn't be here without your efforts, your ideas to use already approved treatments in new ways. And we're grateful for your partnership to really make an impact in patients around the world. So thank you so much. And we're looking forward to working in getting all of these trials funded in the coming months. Thank you.
Thank you.